A DNA molecule consists of two strands and base pairs. Each strand is made out of nucleotides. A nucleotide has three components: a 5-carbon sugar, deoxyribose, a phosphate group, and the nitrogenous bases. The four nitrogenous bases are adenine, thymine, cytosine, and guanine.
Nitrogenous bases make up the building blocks of DNA, basically a molecule that contains nitrogen that has the same chemical properties as a base. Adenine pairs with thymine and cytosine pairs with guanine.
The top strand orientation is 5' to 3', while the bottom strand orientation is from 3' to 5'. In DNA replication, DNA must be replicated in order to ensure that each daughter cell can receive a complete set of chromosomes during cell division.
Helicase unzips DNA by breaking the hydrogen bonds between the base pairs, which creates the replication fork. The replication fork is the area where DNA replication actually takes place. One strand is called the leading strand, and the other strand is called the lagging strand.
The origin of replication is where the replication is happening as helicase unzips the DNA molecule. Sites where nucleotides are exposed, called replication forks, then appear. SSBPs hold the DNA strands from coming back together. They bind to each separate strand, holding them apart.
The unwinding of the helix causes tension further ahead of the chain of base pairs and the strands. Topoisomerase relieves the tension of the chain and breaks bonds.
Primase lays down primer so DNA polymerase can insert new complementary bases.
RNA primer. A short strand of RNA bases creates the enzyme RNA polymerase, which then allows it to build off of the older DNA molecule strand and attach the sugar of one to the phosphate of the next to build a new strand off the pre-existing DNA strand. The new strand that is being built to one of the separate older strands is called the leading strand. The other side of the old DNA strand has to be built upwards from 5' to 3'. The construction is discontinuous and is made up of short segments called Okazaki fragments, which make up the lagging strands.
DNA polymerase binds to the primer and inserts new complementary bases and builds the sugar phosphate backbone. It always goes in the 5' to 3' direction.
DNA ligase seals the Okazaki fragments together.
At the end of DNA replication, there are two new DNA molecules. They are semi-conservative because each DNA molecule is made up of an old strand from the old DNA molecule, and it also has a new strand.